Hi, I'm Dr. Rajiv. Let's start with GLP, that is Good Laboratory Practices. So, why GLP? Why Good Laboratory Practice? The first concept was introduced in New Zealand and Denmark in 1972. Okay, and later in US by FDA in 1978. Why? Because of the industrial bio test lab scandal. Okay, so now what is the function, like why this GLP and why? Because of the poor lab practices, like equipments were not calibrated into the standard form, right? So they were giving the wrong measurements, or we can say inadequate test systems, or incorrect or inaccurate accounts of the lab study. So proper documentation was not there. So because of these things GLP was created. Okay, so it was first introduced in New Zealand and Denmark in 1972, and then due to industrial bio test lab scandal, US FDA approved and started this in the 1978. So basically what we're going to do is our learning objective is to understand the concept of GLP, okay, and to know the importance of standard operating procedures in GLP. Okay, to understand definition and concept of SOP and the protocol. And for now we are leaving this quality assurance and quality control, we'll discuss in a separate part, right? So good laboratory practice is a requirement and necessity for a lab, right, to increase the validity and confidence in the quality data received out of experimental procedure, which must be reliable, consistent and reproducible. So it increases the validity and the confidence in the quality data received out of experimental procedure, which must be reliable, consistent and reproducible. It is defined in the OCED principles. What is OCED? This is Organization for Economic Cooperation and Development. Okay, that is OCED. OCED. What is OCED? It is the Organization for Economic Cooperation and Development. Okay. Now, I think it will be OECD principles, that is Organization for Economic Cooperation and Development. Okay, now it is a quality system concerned with the organizational process and the condition under which non-clinical health and environment safety studies are planned, performed, monitored, recorded, archived and reported. Okay, so it is a quality system concerned with the organizational process and the conditions under which non-clinical health and environment safety studies are planned, performed, monitored, recorded, archived and reported. Okay. Now, the amendment, the good laboratory practices have been made as a law by introducing it as Schedule L1, which is new schedule under the Drug and Cosmetic Rules of 1945. By the Gadget notification of this in 2008 and 10, right? Consequently to this amendment, Rule 74, 78 and 150E of the Drug and Cosmetic Act 1945 have been amended. It involves number of good practices in the quality control labs, which are to be undertaken to carry out analysis with a defined degree of accuracy and precision. So if you say what is the definition of GLP, the GLP it embodies a set of principles that provides a framework within which lab studies are planned, performed, monitored, achieved and reported. So the importance, since raw materials, packaging materials, intermediates and finished products are ultimately released based on analytical results generated in the QC lab, that is the quality control lab, accuracy, precision and reliability of these results are of paramount importance. Okay? So this we have to understand. Now, what is the purpose of GLP? Purpose of GLP, if you say purpose, it is to certify that every step of analysis is valid or not. Right? This is the purpose, that every step of the analysis is valid or not. It assures the quality and integrity of data submitted to FDA in support of the safety of the regulated products. GLP have heavy emphasis on data recording and specimen retention. So maintaining of the documentation is very important. So this is the importance of GLP. Okay. About a history, the birth of GLP we were said in the New Zealand and Denmark. In the early 70s, FDA investigated a number of cases of poor practice in toxicology lab throughout USA. Results of being 40 labs revealed many cases of poorly managed studies, insufficient training of personnel and some cases of deliberate fraud. So here we started. Now what happened, what were there in the findings? Poorly trained study directors and study personnel, poorly designed protocols were not followed, procedures not conducted as prescribed, raw data badly collected, right? Raw data badly collected, not correctly identified, without traceability, not verified or approved by responsible persons, lack of standardized procedures, poor animal husbandry, inadequate of test items and test systems, inadequate resources, equipment not properly calibrated or otherwise qualified, reports not sufficiently verified, inaccurate account of study or raw data, inadequate archives and retrieval process. This were the FDA findings. So where every event there is an reaction. So what was the FDA decision? Was a introduction of a new regulation to cover the non-clinical safety studies. So GLP regulation was drafted in 1976 and enforced in 1979. Right? So GLP is a formal regulation that was created in the 1978. In 1981, an organization known as Organization for Economic Cooperation and Development, that will be OECD was that produced GLP principles that are international standards. Okay. Now, non-US companies that wanted to do business with the US has to comply with the norms of the United States GLP regulation, then only they will act. So what were the objectives? The GLP makes sure that the data submitted are true reflection of the results that are obtained during the study. GLP also makes sure that not to indulge in any fraud activities by the lab. It promotes the international acceptance of the test, and GLP promotes quality and validity of the test data. What is the main goal? Reliable, that obtain results that are reliable, repeatable, auditable, recognized. So there are 3 R's and A. Okay, triple R and A: reliable, repeatable, recognized and auditable. Okay, so it also limit the waste of resources, ensure high quality of results, ensure comparability of results and promote mutual recognition of the results. Now there are 10 principles of GLP. First is the quality assurance program. Next is the apparatus, material, reagents, test systems, test and reference substances, SOPs, facilities, performance of study, right? Reporting of reporting of study results, storage and retention of records, and testing facility, organization and personnel, right? So we'll start with testing facility, organization and personnel. It is basically like it has they should have a knowledge of the GLP principles, access to the study plan and appropriate SOPs, comply with the instructions of the SOPs, record of the raw data, right? Like study personnel are responsible for the quality of their data, exercise health precautions to be minimizable risk, and like ensure the integrity of the study. Second, we'll see this quality assurance program. This quality assurance program, that is the like to assess the updated study plans and SOPs, documented verifications of the compliance of study plan to the GLP principles, inspections to determine compliance of the study with the GLP principles. And there are three types of inspections: quality, there is study based inspection, facility based inspections, and process based inspections, right? Inspection of the final reports for accurate and full description, report the inspection results to the management, and statements, right? Third is the say facilities. Let's discuss facilities: suitable size, construction and location, adequate degree of separation of the different activities, isolation of test systems and individual projects to protect from biological hazards, storage rooms, suitable rooms for the diagnosis, treatment and control of diseases. Okay. Apparatus, materials and reagents. These are apparatus of appropriate design and adequate capacity, documented inspection, cleaning, maintenance and calibration of apparatus and materials not to interfere with the system, test systems, chemicals, reagent and solutions should be labeled to indicate identity, expiry and specific storage instructions, right? Say suppose test system, physical and chemical test system, biological test system will be there, like records of source, date of arrival, and arrival conditions of the test systems should be noted. Cleaning and sanitization of the containers, pest control agents to be documented, and records of source proper identification of test systems in their container when they were removed. Test and reference substances, receipt, handling, sampling and storage, characterization, known stability of test and reference items, stability of the test items in its vehicle or the container, experiments to determine stability in tank mixtures used in the field studies, samples for analytical purposes for each batch. Okay. Now the SOPs, the important one, the written procedures for a laboratory program. They've defined how to carry out protocol specified activities, most often written in a chronological listing of action steps. They are written to explain how the procedures are supposed to work, right? Now performance of the study, prepare the study plan, content of the study plan, identification of the study, records, dates, reference to the test methods, information concerning the sponsor and facility, conduct of the study, right? Reporting of study results, information on sponsor or test facility, experimental starting and completion dates, a quality assurance program statement, description of material and test methods, results, and the storage, right? Now the storage and retention of records, that is the study plan, the raw data, samples, inspection data and master schedules, SOPs, maintenance and calibration data. If any study material is disposed or before expiry, the reasons to be justified and documented, index of materials retained, right? So there were all this GLP regulations and documentations. We have actually studied this. There are 5 basic plans. These were the 5 basic plans this I was talking about: resources, characterizations, rules, results and quality assurance. So resources, we have organization, personnel, facilities and equipments. So we'll discuss one by one. that is resources, so organization and personnel. GLP regulation requires that the structure of R&D organization and the responsibilities of R&D personnel be clearly defined. GLP, okay, and also stresses that there should be sufficient staff to perform the tasks required. The qualifications and training of the staff must also be defined and documented. Facilities and equipment. The regulations emphasize the need for sufficient facilities and equipment to perform the studies. All equipment must be in working order. To ensure this, a strict program of qualification, calibration and maintenance must be adopted. And see, this is the characterization, this test item or article, identification and quality, and test systems like identification and health status. So in the characterization, in order to perform a study correctly, it is essential to know as much as possible about the materials used during the study. For studies that evaluate the properties of pharmaceutical compounds during non-clinical studies, it is prerequisite to have details about the test items and the test system. Often an animal or plant to which the test items is to be administered. Rules, that is the study plans and protocols. Now, protocols are written procedures, the main steps of the research studies, and are prescribed in the study plan or protocol. Being able to repeat studies and obtain similar results in a sine qua non of mutual acceptance of data, and indeed a central tenet of the scientific method. So the details of routine procedures must also be available to the scientists involved in the study. However, the protocol which provides the experimental design and time frame for the study does not contain all the technical detail necessary to conduct the study. These details are found in written standard operating procedure. With the protocol and the SOPs, it should be possible to repeat the study exactly if necessary. Results. Raw data. All studies generate raw data. These are outcome of research and form the basis for establishing scientific interpretation and arriving at conclusions. The raw data must also reflect the procedures and condition of the study. Final report. The study report contains an account of the way in which the study was performed, incorporates the study results and include the scientific interpretation of the data. The report is provided to regulatory authorities as part of the submission for registration and marketing approval. Right? Archives. The storage of records must ensure safekeeping for many years and allow the prompt TA. Quality assurance, yes, it is there in the GCP also. It is defined with the GLP in terms of persons, also called the quality assurance unit, charged with assuring management that GLP compliance has been attained within the laboratory. Right? Now, there are the summary of all the GLPs and related definitions. Okay. Now, the SOP. What are the benefits of the GLP? The benefits of the GLP, like it will be a better image of a company as a quality producer in a global market. It provide hot tips on analysis of data as well as measure uncertainty and perfect record keeping. Provide guidelines for doing testing and measurement in detail. Provides guidelines and better control for maintenance of instruments, environment control, preservation of test records. These are benefits of the GLP we have to understand. Now the SOP and what is the relation of SOP and GLP, we'll see. Like SOP and GLP, a testing facility shall have standard operating procedure in writing, setting forth non-clinical lab study methods are adequate to ensure the quality and integrating of the data generated in the course of the study. So this is important. Each lab area shall have immediately available lab manuals and standard operating procedures relative to the laboratory procedures being performed. Published literature must be used to supplement the SOP. A historical file of SOP and all revisions thereof, including the dates of revision, shall be maintained. So, what are the contents or characteristics of the SOP? This is the like total integration, comprehensive coverage, readability, usability and traceability, understanding, responsibility, change and control, centralized organization, availability and archiving. So these are very important contents of the SOP. So thank you, and we'll start with the like every industry, from like even the building of an industry, the plan and everything starts with GLP. Okay, if we are making an industry, we should know what space should be given to the labs and everything. So GLP is there in all aspect of the pharmaceutical industry. It is there in the microbiology labs, all the labs, in the institutions there are labs. So like they have to follow a particular guidelines, particular standards, and so you should understand and should practice GLP, good lab practices. Okay, so thank you.